All right, everyone, we're going to go ahead and get started. Thank you so much for being here today at our research seminar. My name is Cinnamon Moffett, and I'm the research program manager at Oregon State University's Hatfield Marine Science Center. And you are at our seminar series, and we are so thankful that you're here. This is a hybrid event, so we have folks online and folks in the room. For folks online, if you haven't joined us before, we do have your mics, cameras, and screen shares turned off, but please put any questions you might have or any technical issues you might be having in the chat box. We have a volunteer in the room, Roseanne, who will read those out to our presenter when we're done, or be able to help fix any technical issues you're having. So feel free to use that chat box so you can engage with us. For folks in the room, I'm looking around, most of you have done this with me before, but when you get to the question and answer period, just raise your hand and we'll make sure you get a mic so that folks online can hear your questions as well. I've got a couple of announcements before we get started. I wanted to let you know that we've got an interesting talk that'll be coming in on Monday. So we have a lunch and learn on June 17th in the library seminar room. So you're welcome to bring your lunch, and it'll be at noon. We have Valentine Punatz from Paris Institute of Planetary Physics will be here exploring the largest submarine eruption on Earth, a trip to the Indian Ocean. So I'm hoping that you can come and be a part of that, bring your lunch and join us in the library seminar room on Monday. I also wanted to let you know that next week we will not have a seminar because we'll be hosting our Markham Symposium, which celebrates our students. And so from 2 to 5:30 in this room and in the atrium outside, we'll have a poster session, we'll have a keynote speaker, maybe from somebody you're going to hear a little bit more about today. Brittany Skortskoff will be here. She is a research fisheries biologist with NOAA and a past student here at OSU in Hatfield. And then there'll be some lightning talks from our students. I'm hoping you can come and join us for that. But I'm just going to quickly transition over to today's speaker. Scott Heppell is a professor with the Department of Fisheries, Wildlife, and Conservation Sciences here at Oregon State University. He got his bachelor's from the University of Washington in biology, his master's and PhD from North Carolina State University. And Scott's research focuses around how physiology, behavior, and life history traits affect interactions between fish populations and their respective fisheries. He collaborates with academic scientists, state and federal agencies, foreign agencies and universities, commercial and recreational fishermen, all working together to address the issues related to the sustainable fisheries in marine and freshwater ecosystems. And so I'm just going to quickly hand this over to Scott.
And then for that introduction, hopefully everybody can hear me okay. Is this I can speak like that? It will. Great. So thanks for being here. I should point out that ultimately I'm up here giving this seminar today a bit by mistake. And my mistake was sending a paper to Cinnamon a couple of months ago that we had just published saying, hey, I think some folks at Hatfield might be interested in this. And her response was, great, you're down for seminar on June 13th. So here I am. But really what I want to talk about today is this study, right, Decadal change in the abundance of fishes and invertebrates of Yaquina Bay. But I'm going to tell a little bit more of a story about how that work came about and really look back over several decades worth of work. And I want to acknowledge several people that were really key to making this happen. First of all, Selena Heppell, but Brett Gallagher, Scarlett Arbuckle, Allie Daubel, Amy Jo Lindsley, and then Brittany Schwarzkopf, who Cinnamon just mentioned will be the keynote speaker at the Markham Symposium next week. So I'm going to start off not looking back all the way to the research I'm going to talk about, but really looking back almost 25 years to when Selena and I sort of first washed up on the shores of Newport. And we had actually moved back. I did do my PhD at NC State, but I grew up in Oregon, and Selena was from Seattle, and we were looking really, when we were done there, for the first train back to the East Coast, or back to the West Coast from the East Coast, and we ended up here. And as all new faculty do, right, we have to get together and our work, develop our research programs, become famous, all those sorts of things, meet the departmental teaching needs. But we had a real keen interest in doing one more thing, and that was really working on providing student opportunities that weren't available in the way we felt maybe they could be at OSU. And we were really interested, particularly on the marine side. There was still lots of marine work going on, but we were trying to build that not just here at the Marine Science Center, but make connections on main campus where we were based so that we could encourage students to be down here. And we were interested in doing professional development work, skill building, hands-on opportunities, marine science, and making not just opportunities where they can get out and learn how to do field sampling, but actually meaningfully contribute to some scientific endeavor. And so we ruminated on this for a while. Our department, Fisheries and Wildlife, at the time had a really good model for this called Stream Team. And this Stream Team was something at that time, it's still going on, and so it's now like 60 or 70 years old, is really one of these all-encompassing, in that case, freshwater ecology approaches. And so we said, well, we should develop something like that. And so we developed this organization, this group called Marine Team. And it was open to any and all students, both undergraduate and graduate, although targeting undergraduate students. And we got some very good initial interest. We got them together and we decided, okay, we have a group. Well, now we need something to do. And what we really decided to do, and this is again what I'm going to share with you today, is to really decide to allow not just the opportunity to contribute to meaningful research, but to actually develop that research itself. So that was our fundamental premise. So we kind of batted ideas back and forth for a term or two with the students. And then at some point in time, somehow, and I can't remember the origin story of this, we came across this paper by Wally DeBen and others from 1990. And I can't remember if one of our students found it. I have some vague memory that Ted DeWitt actually handed it to us and said, here's something that went on in Yaquina Bay that might be a good opportunity for students to replicate. And why is that? Well, first of all, it was this multi-year trawl survey of Yaquina Bay. And what they were really interested in is looking at the diversity and abundance of the epibenthic crustaceans and demersal fishes. It was conducted in 1967 and 1968, so a really long time ago and a very fundamentally different time for Yaquina Bay. They had identified specific stations up and down the estuary, all the way from sort of right out in front of the Marine Science Center here, all the way upriver past Toledo. And they sampled it over and over again in these different locations to evaluate broadly, as I said, the diversity, abundance, and fishes of crustaceans in Yaquina Bay. So that in itself was really interesting, but what was really great about this paper, because we had a hard time getting hold of the original data, fortunately in there, they published a map. And that's, of course, where all great adventures begin. And we studied this map. We thought about whether or not we could create a doable project about this, created our own map out of this. And what they had in their own study, let's see if I can get our pointer to work here, is they had identified multiple segments of the bay, sort of a section, segment 1, 2, 3, 4, and then 5 way upriver, and identified specific sampling sites within that. And we took this information, recreated our map, and decided that this is something we could do with students in the early 2000s, approximately 35 years after that original work had been done, and just ask the question, what has changed? So we got ourselves a boat, and we got ourselves a crew, and we started collecting data. Some of you may recognize that boat. That's that poor, sad, languishing boat that's out in the boatyard now, but originally it served us very well for this particular project. And we set up our study as basically a DeBen et al. 2.0, starting in 2003. And the idea was to replicate that 1967-68 survey design. They did biweekly sampling, so we did biweekly sampling. Our time period was sort of mid to late 2003 to the middle of 2005, approximately the same number of months sampled. All the work was done with a 16-foot otter trawl, and we replicated the trawl distances of approximately 1 kilometer, basically trying to control as much as we could, having not been there 35 years ago, to replicate what they did. We focused, as I said, on the lower three sections of the bay. Running the entire bay wasn't feasible with students, with everything else that they have going on in their lives, but we could do a really good job of understanding what's going on in the lower bay. We were most interested in the lower bay anyway, because that's where most of the change had happened in the intervening 35 years as well. Very simple work here. Basically identify, enumerate, measure, release. That's what we did with pretty much everything that we caught. We didn't really retain anything. Occasionally, if we had something very odd, we would retain it for a positive ID. But really, this was doing all those sorts of things that they had done in this original work with some, I guess, slightly more advanced gear, right? We did, in fact, take a variety of water quality parameters as well, salinity, temperature, DO, those sorts of things. But the rest of it was something that was really tractable for undergraduates who were getting their first hands-on experience, first time getting their feet wet and cold out on a boat. Collect things, identify them, measure them. So over and over again, repeat, 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 bi-weekly, in our case for 26 months, I believe. Something like that, close to it, not quite, right? I should point out that while we were collecting length data, there were no length data available to us at the time from the Decadal study. We collected those anyway, but I don't have anything really to report from a comparative perspective on that at this time. But that does bring us to today, which comparison across 35 years. There's the original paper from Decadal in 1990. Ours just came out in April. I want to point out a couple of different things here. First of all, it did, in fact, take us 19 years to publish this work. That's what we would call a long languishing manuscript, I think. But it took 23 years for Decadal, so we're way ahead of the game as far as that's concerned. Second, I think it's great that the journal Fishes, where we published it, thought the work was interesting enough that they wanted to put it as the cover photo for the journal. It's always something we should strive for. And then the third thing, one of the things I sometimes tell my students is that one of their goals in publishing in their scientific career is they need to figure out a way to get their own picture in a scientific publication. And I've just now done that for Marissa Litz, Brett Gallagher, and Caitlin Bosley. So you're welcome. You are now enshrined in the scientific literature. All right. So we're interested in the bay, what's there, and we wanted to put it in the context of what's changed. So this is an aerial photograph. I can't remember the exact date, but I think it was late 1950s, 56, 57. And you should all be able to kind of familiarize yourself with the different areas. You can see the bridge, for example. You can see the dunes mounding on South Beach. You can see a large sandy mudflat right there. We are, where are we? We're like right here, something like that. Lots has changed over time, and we're going to look at it. So this was sort of early on, even before Decadal and his co-workers were here at the EPA lab with the Western Ecology Branch doing all this work. About the time they were doing this work, it looked like this. This is like 1966, 67, I believe, maybe 68 when this photo, right? So Hatfield is starting to emerge from the tidal flats of South Yaquina Bay. But if we really want to look at what's changed, and this is a more modern photo, so this isn't actually 2002, 2000, or 2003 when we were working, but a lot has changed over time that is noteworthy, that affects both available physical habitat, but then of course is also going to pretty substantially affect the hydrology of the system as well. And we'll start with 1969. That is significant because that is the year that the U.S. Army Corps of Engineers changed the dredge depth for the main channel, which starts at the jetty, at the jaws, at the tips of the jetties, and runs all the way up basically close to the turning basin by the liquid natural gas tank. They deepened the main channel from 26 to 30 feet, and I believe the entrance itself went from 26 to 40 feet. So as you can imagine, that might alter tidal flow reasonably substantially as far as saltwater intrusion is concerned. So the 1970s and 1990s, we see the main developments going on around the Hatfield Marine Science Center. In 1972, Army Corps extended the South Jetty by, I believe, 1,800 feet. And again, that's going to affect how water moves, how sediments are retained, all these sorts of things, meeting human needs, of course, but also then probably altering the ecology of the system. In sort of 1974 to 77 is when Embarcadero Marina was built, some improvements to the commercial town docks, Embarcadero itself being put in along the North Shore as well. 1976 is when the original natural gas terminal went in. 1978, Oregon Aqua Farms built along the edge of the southern side of Yaquina Bay. In sort of 78, 79 is when South Beach Marina was built. My dad complained bitterly when that was built because he felt it was taking away the best clamming beds in the entire estuary. And then a little bit more here and there kind of over time. But so yeah, aquarium coming in 92, some water withdrawal, some hardened structure, so on and so forth. So pretty substantial changes though in how water moves through the system and how much available intertidal habitat is available. And I keep talking about that, and I should point out the reason we're interested in it from a fish perspective, of course, is that estuaries serve as reasonably important nursery grounds for a variety of different, both commercially, recreationally, and ecologically important species. Okay. Some environmental changes as well, and now I'm going to actually start looking at the paper and some of the comparisons that we've done, although that previous figure appears in the paper, is that if you look at what's really changed in the estuary, there has been real shifts in salinity and temperature, not as much as oxygen, so it doesn't appear in this table, but things to point out is that on average, sort of the average salinity for a given month across the entire period has shifted towards more salty environment, consistent with what we might expect for more saltwater intrusion with a deeper channel. Not a lot of difference in maximum salinity. We can only get so high, right? 34 to 60, 36 is about where we're going to get when we hit full strength seawater. But particularly when we look at the summer monthly salinity range, we have shifted way up. And in the temperature range, we have shifted way down. So we have a system that is fundamentally different in 2003 to 2005 than it was in 1967 and 1968. So a bit colder in the summer now than it used to be. Interestingly enough, just a touch colder then than it was now, but I've been pondering this a bit, and at least a reasonable explanation to me is that particularly in the wintertime, the ocean is probably, and tidal flow into the estuary, is a moderating effect on temperature relative to cold rain and snow runoff coming off of the land in the wintertime. Whether that's actually true, I don't know, but it intuitively makes sense to me, okay? So some big physical changes, big environmental changes. Now we're going to actually look at the meat of the work, which I said is, again, the work that this great team of students did over a variety of time. And I'm going to look really sort of, if you take the top 95% of the catch, because as we get down towards some of the species at the bottom, there's a lot of just ones caught one of these, one of these, one of these. But if you look at the sort of the majority of species that we were seeing in the system, if you count everything that we caught, right, just on a catch per unit effort, how many individual organisms did you catch per trawl event? It dropped from almost 650 to about 60, about a 91% decrease. And if we're using CPUE as our index of abundance, that's a 91% decrease in abundance. If you just look at epibenthic crustaceans, and here we're almost exclusively talking about shrimp and crab, it's actually increasing though. We actually saw more epibenthic crustaceans in our surveys on a catch per unit effort basis than they did, than Ben and his coworkers did back in 67 and 68. However, if you specifically remove Dungeness crab from those data, we have a pretty substantial decline in the overall abundance of epibenthic crustaceans except Dungeness crab. And if we look at fish all by themselves, we see about a 95% decrease in total abundance of fish on a CPUE basis. Real substantial change, it would seem. So let's look then a little bit more at individual species. And we'll start off with English sole, which was the dominant species in both surveys. Over their sampling period, they caught almost 11,000 English sole. We caught about 1,800. And this is really interesting to me in reflecting on this, because when we were out trawling, I was like, wow, this bay must be paved with English sole. We always catch English sole. 10-minute trawls, we're catching 100 of them or something like that in some places on some times. But they were catching them by the thousands. So it's a little bit of a shifting baseline. I thought there were plenty out there. And it turns out, relative to what they had discovered before, big differences. Okay? And we can go ahead and look on down the rest, but that's an 84% decline in English sole. If we reveal the rest of our list, and this is the top 10 as were caught in 1967 and 1968, we'll highlight a few more. We'll start off with snake prickleback, almost completely disappeared from the system. We caught a few, but not nearly in the numbers that were caught. We caught 23, they caught 3,000. Dungeness crab, up 275%. That's the one increase that we saw out of anything that we looked at. Buffalo sculpin, way down. White sea perch, way down. California bay shrimp, didn't see one. So fundamentally, it would seem the system is very different, or at least the diversity and abundance of species is. And if we want to look overall at species richness and diversity indices, those are also lower now relative to 35 years ago. So total species count, right? Just sort of species richness, if you will. And we're talking about lower, mid, and upper here. I guess I should have mentioned that lower, mid, and upper are referring to sections 1, 2, and 3 from the original Bend studies. So section 1 is the lower bay. Section 2 is sort of Sally's Bend, headed towards River Bend. And section 3 is up around just by about the oyster plant, for those of you that are familiar with the bay. We went from seeing 50 species to 13, 40 species to 28, 36 to 24. So we see the biggest change in that lower part of the bay, which is also where we saw biggest change in physical structure of the system. And if you look at a variety of diversity indices, Simpson's, which is basically a probabilistic, what are the chances that your next sample is going to be the same as this sample, Shannon's, which looks at richness and evenness, and Margolis, which looks at richness, declines overall. Pretty substantial changes in species diversity as well as overall abundance. So to summarize this part of it then, what changes did we see overall? It's a saltier and colder estuary than it used to be, likely because of greater saltwater access because of changes in channelization. 91% decline in overall demersal species CPUE, but a 9-fold increase in Dungeness crab CPUE. Declines in multiple measures of biodiversity, and a shift in species dominance overall. So this system used to be fish dominated, and that shift is the point now where it is in fact dominated by epibenthic crustaceans, and Dungeness crab specifically. That's what's changed. Big problem, of course, for all this is that we don't have any, it's not an experiment, right? We can't manipulate things, so we can't really get at causal mechanisms. Now, I've put forth what I am, have reasonable confidence that is linked a major contributor to it, which is physical and environmental changes to the system. But it's probably important to look at some other explanatory variables, which I don't think we'll ever really be able to answer. So it'll be a great conversation over a beer at some point in time, and we can debate these. Could just be random environmental variability, right? That can't be discounted. Maybe they were just sampling in a really, really good year and we just sampled in a really poor one. I would suggest that a 91% decline is more than just a good year versus a bad year, but maybe. Chris Langdon rightfully pointed out that maybe there's something to do with changes in marine mammals in the bay, which have certainly changed a lot as well. And we'll look at a figure of that here in just a second. Maybe there's a lot more predation in the system on some of these animals. And that's certainly possible. And it's not that it can't be a contributing factor. I think that considering the declines we see across the board and the fact that the pinnipeds in particular, what we're talking about here, probably aren't feeding across that range of species, but maybe they are. Similarly, changes in piscivorous bird populations, cormorants, loons, all those other sorts of things out there. Climate change could be a big one, right? Climate itself overall is very different when the timing of upwelling is happening, changes in pH, all these other sorts of things. And then the other one that I think is interesting to ponder, and I'm going to give some more evidence for this, is that over that time, we've also seen some changes in conservation and management that may have been one of the causative factors for a low point for some of the things that we were seeing that were in decline in the bay. So I'm going to look at a couple of these just to give us something to noodle on. Dungeness crab in our sampling has going up. But what's interesting is, of course, is that Dungeness crab harvest on the Oregon coast has a lot of variability to it, but is also going up. That's what it was at in sort of 1967, 1968 during the Decadal study. And that's where we're at during our study, right? Again, lots of variability, but on average, increasing catch. Increasing catch, if we're using that as with, of course, increasing effort, but if there are more adults out there, they may be giving rise, and most of the crab we're sampling were pretty small. We may be seeing more of those individuals in the bay simply because there's more out there. Another part of sort of the human user side of things is that there were in the over the years, and particularly in the year 2000, sort of 2003, pretty substantial changes in the management structure of groundfish on the US West Coast, the groundfish disaster declaration, changes in management, and that was all led by a variety of stock assessments that demonstrated that some populations, particularly of rockfish, were in decline and something needed to be done. And so Decadel's study was done, when I'm using rockfish as a proxy here, but this might be true for some other species as well. When populations were really hot, reasonably high, our study was done here, right? So when some populations anyway were depressed. All right, and here's that marine mammal slide. It's hard to find marine mammal data prior to 1975. This is from NOAA. Of course, that's when, about the time Marine Mammal Protection Act went into place. And Decadel's study is way there off to the left. It doesn't even appear on this graph, but we would assume marine mammals low. And relative to when we were sampling in 2003 to 2005, at that point in time, this is for California sea lion populations, had been considered recovered to above their target threshold. So there's certainly some credence to what Chris brought up. And so I think that requires some more investigation at some point in time. Okay. I'm going to transition a little bit now and talk for a few minutes about where we've gone since that time with some more focused studies about understanding the bay itself, the importance of different habitat types, and how that plays a role in habitat areas of particular concern. Come talk about seagrasses, essential fish habitat more broadly, and the role of estuaries. And this is going to cover, to some extent, the graduate work of Brett Gallagher, Allie Dauble, Amy Jo Lindsley, and Brittany Schwarzkopf. So here's hoping this isn't what Brittany's going to talk about next week. Okay. And in fact, these are going to cover these 4 papers, primarily, which Brittany was the lead on 3 of these, and then Allie was the lead on the 4th. And overall, as I'd already mentioned, there have been these declines in a variety of different species of rockfish and some non-rockfish species as well. They had been declared in that overfished zone. And so overall, one would have the impression anyway that rockfish populations are lower than maybe they should be, and certainly lower than they used to be. And so a lot of the work that we had done since then was really getting at recruitment, settlement, growth, diet, survival of estuarine fishes, primarily in natural habitat seagrass, but we were really interested in how they performed in other habitats as well. And so when Brett was sampling in 2004, 2005, he sampled a lot and he caught a lot of rockfish. But every single rockfish he caught was a black or a yellowtail. And they were like 96% black rockfish. So Since Brett's time, when he was sampling, every single one of those stocks has been declared recovered. Right? And so that would imply an increase in overall rockfish populations out in the ocean, producing more babies, maybe better recruitment, and perhaps more diversity of recruitment as well. And there was lots of news about it out there, right? West Coast rockfish. In fact, they recovered quite well and quite fast because of actions of the fishing fleet, actions of the management councils, lots of different things. And so DeBen's work was done there, ours was done here, but that's what's happened to those populations since then. So as we move forward to Allie's work, Amy Jo's work, and Brittany's work, there's Brett there, Allie's work there, Amy Jo's work there, and Brittany's work there, we start to see a fundamental change in diversity of recruitment in the estuary. And again, we're focused on rockfish, probably other things going on as well. When Allie was working in 2008, 2009, she saw 3 different species. And we did the genetic ID work with Carlos Garza down at Southwest Fisheries Science Center. So thank you, Carlos. By 2012, 2013, for Amy Jo's work, she was seeing 8 different species. And for Brittany's work, we were seeing 10. We were seeing more and more species recruited into this estuary, which is kind of cool. Don't really have an explanation for it other than that speculation, but more. And that's what we see for them coming into the estuary. I've already talked about the development, what we call hardened structures being put in the estuary that have a variety of different impacts. And we were really interested in the role that hardened structure versus natural habitats play, again, in the growth, the settlement, the growth, survival of juvenile rockfish. If you look at the entire Yaquina estuary up to the head of tide, there's about 125 total shoreline miles, of which only about 11% are armored, right? Hardened in some way, riprap, seawalls, pilings, those sorts of things. But if you look at the lower estuary, the lower 7 miles, right, down near the mouth and the part of the bay that we're standing in right now, almost 94% of it is hardened. And so there's lots of hardened structure around here, anthropogenic habitats. And we've been really interested in, well, what role does that play relative to channel dredging, relative to natural habitats? And so part of Allie's work, Allie Dobble's work, was to look, in fact, she went beyond Yaquina Bay and she looked at a variety of different estuaries up and down the coast, from Nehalem in the north to Coquille in the south, and looked at the total hardened linear shoreline, that's HLS, the second to last column on the right there, and MCA, which is the mouth cross-sectional area. So basically, how big is the opening that is spilling out into the ocean? And different estuaries, according to DLCD, are categorized as deep draft, conservation, shallow draft. And so she had paired 2 deep draft, 2 shallow draft, 2 conservation estuaries, and looked at the role of hardened linear shoreline and mouth cross-sectional area in relation to how much we get recruiting into the estuary. And it's actually really quite interesting that there's a positive relationship for, on the left, total linear shoreline, and on the right, cross-sectional area. And keep in mind, those two are autocorrelated. The bigger the cross-sectional area, the more fish come into an estuary, right? So they're basically, there's more water flow in and out, and so they're easier for them to be advected into this system. So that would mean that even though, say, Siletz, which is this nice, what we consider to be somewhat natural estuary, has fewer fish recruiting into it relative to the industrialized Yaquina or Coos, mostly because there's just more water flow probably moving in and out. And in fact, that positive correlation says for each increase of cross-sectional area, 1 kilometer increase of cross-sectional area, we get about 30% more rockfish coming in. And with hardened linear shoreline, we get about 20% per extra kilometer, right? So that's how many fish are coming in. The Brittany's work then was to look at, okay, well, what happens once they're here? Just because they make it in, is that an ecological dead end? Do they become sort of, ecological ghosts? Or is, once they're in, they're good regardless of where they settle into their habitat? And so she was really interested in the relative role of anthropogenic versus natural habitats. And the first thing that she found was that, first of all, there's high variability. This is from her work in 2016, 2017, and sort of total abundance of harvest across months. In 2016, we got a lot more than 2017, so there is high variability in that way. Two to threefold increase in 1 year. That's just, we think, interannual variability. But one of the things we see is that we tend to see more fish once they're in the estuary settling into natural habitats. Associated with that, then, is that the diet, and I won't interpret this entire table, but the diet is much more diverse and we think more calorie rich if you settle into seagrass as well. A diverse diet that's calorie rich likely leads to greater growth rates, which was the next question that Brittany was interested in asking. And so she went through and she did basically marginal increment growth analysis in the otoliths of juvenile rockfish. And in 2016, that year that we saw lots of fish recruiting into the estuary, which she characterized as a good ocean year as far as recruitment is concerned, and I forget the exact parameters she used for that. But what this graph here is showing is sort of the mean recent growth rate in micrometers, because it's an otolith, for fish that she sampled from dock habitats, pilings, things like that, versus eelgrass. And there's no difference. But when she did the same analysis in 2017, that poor recruitment year, which was also what she determined to be a poor ocean year, they diverged. And there was much higher growth rates for animals living in natural habitats relative to anthropogenic ones, as shown there. And ultimately, beyond rockfish, which is what she was really interested in, she looked at how across all of those years of sampling from Amy Jo's work, from Allie's work, and from her work, how much the physical parameters of the system vary in each year. And I really won't get into all the differences here, but that's that 2016, 2017, different 2016 in purple, 2016 purple, 2017 in, 2017 in purple, 2016 in green. Read my own slide. All right. And then she went through and she looked at how across all those data streams, because we were collecting information not just on rockfish of all species, of how far, given that climate variability, does the community composition vary in natural versus anthropogenic habitats. And so what you're going to see here is a walk from 2008 to 2009, 2009 to 2012, all the way up through the data set that we have. On the right is our seagrass in green, on the left is our dock pilings in purple, and basically the length of a step tells you how much a community is changing. And if you start down in 2008, down there, the 2008, 2009, big differences in changes in natural habitat, or less change in natural habitat relative to the anthropogenic habitats. And overall, what we see is very little change in natural habitats year after year after year under the face of substantial changes in variability. And ultimately, that story tells us that community structure vary between habitats and over time, but there's greater community stability in those natural habitats. I wrote a little poem for it. You can read it if you like. But really what this is telling us is that those natural habitats, maybe in a good ocean year, doesn't matter. But in poor ocean years, natural habitats become really important for community stability and growth of juvenile fish in nursery habitats. Given that we are anticipating more poor ocean years as we move forward into the future, these natural habitats will take on an outsized importance for long-term sustainability of some of these communities. Okay. And it also matters because we're rapidly losing seagrass, right? Seagrass is in the groundfish fishery management plan habitat area of particular concern. So we actually need to manage for seagrass in these systems. They're disappearing rapidly globally. They're disappearing rapidly here in Oregon and in California and other parts of the world, up in the Sailor Sea and the Gulf Islands. And our mitigation efforts seem to be a mixed bag, right? And so it would seem that trying to figure out ways to maintain these natural seagrass beds becomes really important. All right. Certainly, as I talk about these things and restoring natural habitats, we're not going to get back to a Yaquina Bay or an any bay. to what it used to be. But we might want to think about ways to minimize impacts or restore systems in ways that allow them to be effective functioning natural systems, right? As we move forward, here's the bay in 1950. Here it is more or less today. And in fact, if we look at our relative to 2003 picture, there's still been a few changes, right? We've got the NOAA Mach-P system going in. A dynamic revetment was built along the eastern shore of Hatfield in 2011. There's been some changes in the international terminal more recently. There's some ongoing work along the waterfront. These are pretty small changes relative to the big changes that happened between the late 60s and the early 2000s, but they're changes. Ongoing dredging going on as well. And in fact, it looks like there's potentially some more changes coming, and I'm not very well versed on this, but found this while I was preparing for this seminar, is that it looks like there's a proposal out there to change dredge channels as they primarily lead into the commercial docks as well. So not sure where that'll go. Maybe one up here. This is in fact a picture that gets generated when you go into Adobe Firefly AI and tell it to give you an aerial photo of futuristic Yaquina Bay. Don't think we'll get there either, but the bay keeps changing, and it's one of these situations where ultimately we, society, will need to decide maybe what we want it to look like. So let me wrap up with some closing thoughts then. There is a complex community of fishes out there. That community has changed drastically. Not sure yet why, but it definitely has changed. The Yaquina Bay is legally designated as an essential fish habitat. In fact, and the seagrass within it is a habitat area of particular concern. So we do have reasons to want to worry about such things. We also think that then for this part of the study that the natural habitat likely makes for a better nursery, more diverse diets in those natural habitats, more stability in those natural habitats as well. As I pointed out, habitat choice matters in variable ocean years, maybe not so much in good years, maybe it doesn't matter. In poor, maybe there's enough to eat everywhere. In poor years, it really matters where you settle out, how well you do, right? So really, where are we going to try to head next with this? Well, my first goal, now that we've worked through this, is to do another trawl survey. We're 20 years on from that, right? So I'm figuring 20 years from now, I'll be able to publish this. System continues to be manipulated, right? We see all these different changes to meet human needs, among other things. Climate continues to change. Management conservation efforts have changed as well. So that's one of the things I really want to do. And then to highlight some upcoming work that a couple of my PhD students are going to be doing. Olivia Boyson is going to be working not just on understanding these natural habitats and their role in the lives of fish, but how effective seagrass restoration is in mimicking those natural systems, and to some extent, how long do we have to wait for a restoration effort to actually provide those same services that we see in natural systems. And then partnered with that, my PhD student, Wave Moretto, is going to be comparing estuary nurseries to the nearshore rocky reef kelp nursery habitats to see the relative role of estuaries versus those nearshore environments in the growth, survival, and productivity of those populations. I think that's my last slide. I will stop there. Thank you very much for listening. And I'd be happy to take questions.
All right, I think we had lots of opportunity to ask some questions. And so I'm going to bounce back and forth between folks online and folks in the room. And it looks like Roseanne has our first question. Go ahead.
Did the original study give a lot of detail on the sampling gear, thinking about possible differences between nets, actual towing, sampling methods, etc.?
Yeah, that's an excellent question. Can they hear that online, or do I need to repeat that?
No, they can hear it because she was on mic.
Great, thanks. Yeah, so there was some detail, right? 16-foot otter trawl, we knew the door size, we knew the bag liner size. We didn't necessarily know the leading mesh size on the leading edge of the net and things like that. We knew sort of the weight of the net, we knew their tow speed, and we knew the distance covered. So that's what we were able to replicate as best we could.
All right, questions in the room.
I'll follow that question. I spent my life dealing with trawls. I'm amazed you don't have shad in your thing. You don't have herring. these species are evading your trawl, and they're species that are still pretty abundant in this bay. While I think the major factor in here that's caused so much of your species is marine mammals, and the way they've increased in population and the way they hunt is changed from what there were when there was just a few animals here now.
Right. Yeah. So definitely all that true. I wasn't trying to say that it wasn't marine mammals. In fact, like I said, Chris put me onto that, and I'm thinking about that a little bit more. As far as other things in the bay, yes. I mean, essentially what you're asking about is gear selectivity, things you can and cannot catch. Had they used an entirely different sampling gear, purse seine, gill net, beach seine, whatever, and we tried to compare our trawl to that, I wouldn't have much confidence. But since we were trying to compare like to like, that gives us the opportunity to hopefully look at the changes relative to what's there using the same gear, as opposed to trying to representatively sample everything in the bay. And yes, I caught a lot of herring this summer or this spring. So yes, there's definitely a lot of some things around still. But for example, it's hard to see starry flounder anymore, among other things, right? So some things have not disappeared, but are rare in the system now too.
All right, questions in the room, hang on. You raised your hand, my friend.
I was just gonna say the starry flounder thing. I seen all these estuaries for 10 straight years. And so the ALC compared to the Yaquina, deep draft, shallow draft conservation, we weren't seeing starry flounder, maybe a rarity there, similar here. So it wasn't just a change there. That's a whole different factor, most likely perhaps the marine mammal thing, because that's exploded and they eat a variety of things. But as Terry also mentioned, from the seining perspective, when I did all this seining for 10 straight years in set spots, the amount of fish, thousands and thousands of different types of forage fish that you would get, and a lot of sebastes and different things, but a different type, which to me, it's still fish dominant in a way. But I think if you do the study again, you're also going to get a bunch of green crab up in the bay, because we're getting them all the way up to almost Toledo. And Dungeness crab, I caught all the way up to Cannon Quarry. So that tidal influence definitely is a difference between the two bays and whatnot.
Right. And my understanding is that well, I don't know if it's my understanding. I think I've heard that the distribution of Dungeness crab, particularly juveniles, may have moved further up river as a habitat partition from green crab. No? Okay. Yeah. So I've heard from enough people on the starry flounder side of things that that really may be a marine mammal specific thing. And I'm inclined to believe that based on what I've been told. Yeah. Yeah, I mean, ideally we would use every sampling gear and get the holistic perspective of everything. But again, we were trying to compare like to like in this case.
All right, question online. Go ahead, Roseanne.
Great presentation, and so glad you pulled it all together. How much biomass are the trawl species relative to benthic invertebrates? Much more or less, or any idea? Could predation by Dungeness crab recruits affect the biomass of other epibenthic species?
So maybe. Certainly, particularly benthic and benthic infaunal species could be affected. I would totally defer to other people in the room to answer that part of the question. We don't have an estimate of biomass. I suppose we could take our length distributions and try to convert those, but we wouldn't have a way to compare those because we don't have that information from the original study.
All right, questions in the room.
Great study and excellent presentation. 19 years to publish. I'm not too, sorry to hear that, but a couple of things. I mean, obviously marine mammals are an important thing, right? I mean, there used to be a bounty on harbor seals, for example. That's long since changed, and that certainly needs to be looked at. My real question for you is that this is obviously just a fabulous thing to be able to compare over those decades. And I know that Ally looked at a number of different, estuaries, but is there anything from any of the other estuaries that's back that far in time so that it could be expanded to look at any of the others?
Yeah. As far as I can tell at this time, the answer is no. I don't have good access to any agency gray literature that may exist where those surveys could have been done, but were never published or even put into technical reports. So possibly. But in the work that we did in putting this manuscript together, we found one other study from the East Coast that was able to do this broad a comparison, and they found pretty similar responses.
Other questions in the room? Yeah, I'll come back to you, Terry.
So there was one species of rockfish that bothers me that wasn't mentioned. It's what they called blue at one time, but it's now deacons.
Mhm.
I see them in the bay, particularly blues when I was a kid
Uh-huh.
in the bays around the pilings. And I see very few deacons now, but did you detect any deacons, or were you even looking for that species?
Yeah, we were not. And as I recall, our blue catch was extremely low overall. I didn't talk about it today, but part of Brett Gallagher's work is that he did the same trapping work in the bay and on the nearshore rocky reefs, and he would catch blues out there, but not in the bay. And so I'm not sure where that leads us, but it could be another issue of gear selectivity as well, that they could be there and we weren't sampling them.
Well, my experience, the only place I really saw them was in places where there were pilings. One of the changes you didn't detect was in the old days, the docks in the commercial side were made of wood
Mhm.
and had a lot of growth and grass underneath it. And the blues were either around the pilings or
Ah,
directly under that grass. And of course, it was shaded. Today, you have cement docks
the
that
concrete and
don't
styrofoam.
accumulate as much
Interesting.
growth. So there may
Yeah.
be a factor in what these
Mhm.
docks produce in
Right.
rockfish and what was there.
Yeah, that's really interesting. I hadn't thought about that, right? So I mean, I've been focused on structure, but the nature of that structure could play a role as well. That's a good idea.
The other area you didn't cover, and I'm not trying to pick your work apart, it's better than anything else we have. But during a time when we have low oxygen conditions,
Mhm.
a lot of crab move into the bay
Mhm.
because there seems to be better oxygen in shallow water. So that can make a huge difference in your crab populations.
Right.
And also in those conditions, I've seen black cod as far inland as River Bend.
Wow. Okay.
And I've only caught 4 or 5 there,
Uh-huh. But still
but it was in that year of
really low.
low oxygen.
Yeah.
And some species are just trying to get out of the low oxygen, I think.
Yeah, for sure. That's really interesting. I would have to go back. We didn't detect low oxygen where we were working, but as you pointed out, that's why animals are moving in, because of the low oxygen offshore. I would have to go back and ask Francis or somebody else about whether we have good oxygen information for this sort of nearshore environment during the time that we were sampling.
All right, question online. Go ahead, Roseanne.
Okay, I have a comment and then a question. The comment is, sounds like we're ready to get that whaler running again.
Anybody got an outboard motor?
And the question is, has there been any recent research on how aerial herbicide spray affect aquatic vegetation in estuaries? I've noticed a significant decline of aquatic vegetation in the Beaver Creek since they started spraying over the area.
Interesting question. Unfortunately, I have no idea. I don't know if anybody's working on that. So I guess, I'm sorry, can't answer that one.
All right, I saw a couple more questions in the room. Hang on.
Hi, Scott. I had a question on the walk you took us on how the dock and the grass have changed. And one thing you didn't put out on that graph, but Is it the case that in 0708 the dock and the grass were essentially the same? Can we interpret that NMDS that way?
They were, yeah, they diverged over time,
So
right?
at some point,
Not only-
those
yeah.
habitats were very similar
At one point in time.
in species, and they've just
Yeah,
diverged.
that's how I would interpret that. And as we had advanced over time, not only did they diverge, but the docks were moving much faster
Diverged
than
far.
the natural habitat.
So maybe Terry actually gave us the answer to that, is that the docks used to have grass around them and
Sure.
now they don't. So thanks, Terry.
Yeah.
answered
Do you
my question.
know when those dock replacements were done, kind of ballpark time period?
Anybody in the room? We've got homework.
Yeah, kind of curious about that. That'd be interesting to try to work back to there.
All right. Any other questions in the room? Any other questions online, Roseanne? All right. Thank you very much for being tricked into giving a seminar. I really
Thank you, everybody.
appreciate it, everybody. I hope you all come back and join us next week for our student presentations. Thanks, everyone. Thanks, everyone online. We're going to end for now. Hope to see you next week.